Lectures will be given by Stephen again. So, are you okay? Yeah? Okay, excellent. The floor is yours.
Okay. Right, okay. This time I kept the original title, like is in the leaflet. I added a subtitle, but that should be allowed, I guess. I think if you remember my previous talk about use of breeding value estimation, what comes back and will also come back in the next talk on genomic selection is the importance of phenotypic data analysis. And that's why I've been so bold to even call it the foundation of genetic progress. Because in my point of view, phenotypic data analysis has been overlooked in many, many cases. I see companies and institutes doing big investments to introduce genomic selection, genomic prediction, but they don't do any effort in terms of phenotypic data analysis. They take the average over trials or something very easy and continue with the molecular part. Well, I can tell you those kind of projects either completely fail or are at least at a very reduced efficiency, because accurate phenotypic data analysis is for breeding value estimation, GWAS, for all technologies is of crucial importance. Now, if we look at breeding data, what I'll call the breeding data life cycle, most breeding companies and breeding institutes will work with a phase where they will collect data. I call it here phenotypes, pedigree information, molecular data. We'll go on to storing this data somewhere. I've put a database here, but it could equally be just Excel files. You run the analysis, different types of analysis to calculate breeding values, GWAS, whatsoever. And then the crucial part, reporting and decision making. So we're going to focus now in this part on the data analysis thing, and more specifically, phenotypic data analysis. I've noticed that my vision and strategy, it should be centered. So imagine that it's centered. So phenotypic data analysis, very, very often I get confronted with data that is already existing. I call it historic data or data archaeology, as shown here, where there's very little you can still do on the trial design that was used and the way the observations were met. Now, data archaeology is a lot harder than designing new experiments. Well, in the exceptional case that everything, of course, has been done correctly and according to the state of the art, then data archaeology is the most easiest part. But in most cases, working on historic data is quite challenging due to several reasons, but usually incomplete data, suboptimal trial designs, missing data very often, because if a trial is not that good, it is often not imported into the database system and things like that. So of course it's nice if you can be involved from the design of the experiment, looking at what is the sample size, how many replications do we need? Do we need replications? If we use these kind of replications, what is called a power analysis, what is the discriminating power of this trial? Discriminating power means if I put two varieties in there and the one is better, A is better than B, will I effectively notice that A is better than B? Because if they're close together and our trial is not very accurate, it will look like A and B perform exactly the same. So power analysis is basically saying how much effort do I have to put into to be able to distinguish good from bad, in very relative terms, of course. And then trial designs, there's a part on trial designs and the evolution of trial designs and the use of this. Going on to the actual analysis point as well, and this is a very, very broad field, which I can only light a few parts of, the most interesting part in my point of view. And as I've mentioned also in previous speech, digital phenotyping has become something of great importance for getting accurate and objective phenotypes of course, applicable in only certain cases, but then again opens an entire different arsenals of tools and methodologies that are needed to extract features, phenotypes, from these images. So what this slide is trying to show you, that phenotypic data analysis, although it seems like something that most people have learned somewhere in their academic trajectory, have learned to do an analysis of variance, there is quite a big world out there, and the key is to have, from the design to the decision-making phase, to make the right decisions that are appropriate for the problem you're trying to solve. So, if we look at the initial state, field trial design, that on its own is actually a profession. There are statisticians whose job is creating field trial designs. So it's not something that is to be taken lightly. Although in several institutes you often encounter we're going to do a randomized complete block design with X replications because that's how we've been doing it the last 20 years. Well, that might be fine, I don't know, it's not up to me to criticize, but just showing you that there is an entire world of knowledge on field trial design that is often, but not always, underlooked. So what is the goal of field trial designs? I think most of you know, but the idea is to distinguish the effects of the treatments. For plant breeders, treatments are basically the accessions, the varieties, your breeding pool, from the effect of nuisance parameters. Nuisance parameters is basically everything that is not interesting, meaning the effect of the plot, the effect of the row, the effect of the column, the effect of the replication, of the trial, of the environment. All of these things generally are not of interest. You just want to know the effect of your accession. So your trial design is an attempt to try to distinguish genetic effect from all the rest. That is the goal. And in that same goal, try to compare your treatments under uniform conditions. Meaning, if one treatment, again, treatment is accession, is in a very good part of the field, and the other one is in a very bad part of the field, whatever that means, you're not comparing them under uniform conditions. So again, trial design tries to give you as much as opportunity to test your treatments under uniform conditions. Now, what is less known, although I think it's been clear now during the UCLAC project, that the design that you choose for your trial very much determine the availability of analysis methods that you will be using afterwards. So, when you're considering certain trial designs, you're actually already implicitly deciding on how you will analyze it later on. So what I'll call simple, which sounds negative, but basically what I call traditional designs, like the randomized complete block design, there's nothing wrong with that, just to be perfectly clear. In some situations, it's the best possible design that you could do. But in most other situations, it's suboptimal trial efficiency depending what the goal is that you're trying to do. The goal is usually to maximize the number of treatments, accessions that you want to screen versus the number of plots, the surface that you have available. So if that is your definition of trial efficiency, randomized complete block designs often do not win that battle. But if you use, it's not only randomized complete block design, but basically if you use simple balanced designs, meaning that every accession has the same number of plots on your trial, then you can make use of the standard analysis toolbox, which I guess most of you have seen in some statistics course, like the analysis of variance, which afterwards you can do pairwise comparison between your treatments. I've put it here just because it's popular. I'm not a fan, but it's the least square difference, which is still used very much instead of more appropriate tests like the Tukey's test. But basically, anybody who has followed some introductory course to statistics has learned these basic methods, which are very powerful and moreover very robust, meaning that if your trial data does not fit certain assumptions, like in terms of normal distribution and so on, the analysis of variance is known to still be performing quite well, give you quite accurate results. So it is for this reason only that you could opt for using a traditional design, because the analysis afterwards will be so much more simpler. For example, if you calculate the mean, if you have a completely balanced design, meaning that every accession is present on the same number of plots, randomly distributed over your trial, and you take the average of the observations that you make, then that is already a pretty good estimator. It will be hard to correct, to get a better estimate using more advanced models, not taking into account spatial effect, admittedly, but just saying that there's nothing wrong calculating an average as long as your data is balanced, as long as you have multiple replications. So simple designs might be more advantageous if you're looking into more standard analysis. Now, if you're looking at more advanced, and I called / parsimonious designs, parsimonious, again, making the trade-off between the surface that you're taking and the number of accessions that you can test on them, then you're looking at designs of optimal or near optimal efficiency, and in almost every case, you'll end up in the linear mixed model analysis world, which has been talked about several times by me, but also by the other speakers during this workshop. Now, in this linear mixed model analysis, what I've noticed that it is not that known by people that follow an introductory course to statistics. All of a sudden, you don't really have a pairwise comparison test between treatments. A lot of things that you're used to looking at, the QQ plot, quantile plot between your residuals to check if there's a normal distribution. Well, all of these things are, well, you could do it for linear mixed model analysis, but it just wouldn't make a lot of sense to do it because it's very hard to do diagnostics on a linear mixed model residuals. So you're in a completely different ballpark. Now, so you have to decide beforehand, am I willing to take the jump here? Now, what I'm trying to do here, so the goal of this presentation, is trying to convince you that it's worth the effort. So yes, it's a little bit more complicated, but on the other hand, there are several advantages, not only related to the fact that you can use more parsimonious trial designs, but there are other advantages specifically for breeders that are very interesting to have. Now, going back to the randomized complete block design, like I've said, it's a bit the standard design for field trials, and you will try to, so treatments are accessions, I've said it several times, so you will randomly distribute your treatments over blocks. Now, one thing, so you can use, like I've said, the well-known analysis of variance, post hoc tests. So post hoc test, that's for when you want to do pairwise comparison. I want to compare treatment 1 to treatment 2 and get a statistical evidence if they are different or not. That is what often is done so fairly robust to modest violations of the assumptions, because it kind of depends what you mean on a violation of an assumption. So it's robust against, imagine that your variances are not really originating from a normal distribution. It has been shown that all of these tests, like the post hoc test, still provide good p-values. However, if you have different residual variances, for example, per treatment or per block, that would be a gross violation of assumptions, and then it wouldn't count. So it's very relative, but anyhow, it allows for missing value imputation. Again, something I really don't like people doing. But technically, if you have a missing value, what you often see that people will use the least square estimate and fill that in. I'm not saying that's a good approach, but at least it allows you to continue for certain analysis. Randomized complete block design is basically a kind of a spatial structure in one dimension, meaning you will correct for the block effect. So imagine this is the field. I only have 4 accessions to test, so I'm a really lucky breeder. I'll have 4 accessions to choose from. Now, if I lay out my blocks like this, it presumes that there is a kind of gradient in the field, in fertility or shade, and in an environmental condition that goes from left to right, or vice versa. So I'm correcting here for that gradient. Of course, if there's a gradient in this direction, the design does not help whatsoever. Right? So it's a one-dimensional design in that perspective. and it ignores within block spatial heterogeneity. So imagine that this all of a sudden would be a very advantageous place to be. There's no way that you will capture this with the randomized complete block design. And is not very parsimonious in the number of plots. But again, it's a bit of the standard, so it's kind of the benchmark where you compete the other designs with. So going on to incomplete block designs, the idea is a bit similar. We still have big replications. So previously I had 5 replications, quite a lot. So we still have, I don't know how many replications here, but the idea is that I separate each of these bigger blocks into small subblocks. Right, so this is a subblock, an incomplete block. In the first incomplete block, there are only 3 accessions. In the second one, and in the third one. And that is my entire replication. Now, what is different? First of all, you see my blocks. If you look at the individual number 1, treatment number 1, it is in this replication here, it is combined in an incomplete block with number 2 and number 3. In the following replication, this is this zone here, it is together in an incomplete block with 4 and 7. And then it goes on. 1 is together with 5 and 9. 1 is together with 6 and 8. So you see that every pair of individual is combined exactly once in the incomplete block. So they're all very close together in the same environment, because remember, that was the goal of this design, to be able to compare treatments in the same environment, under the same conditions. So this is balanced because every individual is once together in an incomplete block with each other individual. Does it help in terms of parsimoniously? Not at all. As you see, I use the same number of plots. It's just a little bit more intelligent to structure it. This is actually a bad example, as you see, because 1 is always on the same position. It's just to make the example a little bit more clear. It should have obviously be randomized within the incomplete blocks. But you see that the goal is here, so to have the accessions together. Now, this is not very popular because it actually looks very similar to a randomized complete block design in terms of a number of plots that you used. But it's basically an intro to what is known as partially unbalanced incomplete block designs. Not a very catchy name for the family of designs. But the key is that now all of a sudden not all pairs of treatments are compared the same number of times. So this should be a D. You see that all of a sudden we use few less blocks, and we see 1 combined with 2 and 3, and with 4 and 7, but that's about it. 1 will never be together with 9 or 8, and so on. So here, I'm already willing to sacrifice my comparisons. So if I would calculate a comparison, so a comparison is basically just the difference between the mean performance of 1 with the mean performance of 2, the standard error of this comparison would be lower than a comparison of 1 with 9, for example. Because 1 and 9 have never been together in the same incomplete block. So the comparison of their means will have a higher standard error. Which is bad, by the way, if you're wondering. So you want low standard errors, but you are willing to do this sacrifice because it saves you on plots. Now, one of the very successful designs, incomplete block designs, has been the alpha design. I think it's already from the late 70s. You see an example here. It uses full replicates, so it's again a bit similar to a randomized complete block design, but it's very much balanced. It uses these incomplete blocks and tries to balance as much as possible the presence of one, for example, that each accession is always present in an incomplete block somewhere else, but also takes into account that it takes into account the incomplete blocks in one dimension. So you basically have a kind of effect here in this direction, if I'm in this block, or in this incomplete block, or in this incomplete block. And it has been shown to be quite flexible if you have different number of treatments and the number of plots that you have available. You have software that can easily calculate an alpha design. It's not 100% proof, but usually you can find a solution that fits your need using an alpha design. You have full replicates, so if all things go bad, you can still return to a classic analysis of variance. So it's actually kind of an improved randomized complete block design. But one disadvantage, of course, is that it only corrects in one dimension again. Namely, in this dimension here of this block, so here the gradient here would go from left to right or vice versa. So a kind of an better approach would be the row-column design, where you have incomplete blocks in two dimensions, in rows and in columns. So you have full replicates, as I hope you can see here. But in each replicate, you have a kind of an incomplete block in the direction of the column and an incomplete block in the dimension of the rows. And you can actually then correct for the effect of the replication, for the big block, correct for the effect of the column, the incomplete column, and correct for the effect of the incomplete row. So it's a kind of an improved version of this alpha design. And it's known as row-column designs. So each of the time you add a little bit more in complexity, but it allows you to find spatial variation in either row or column direction. Federer's augmented design doesn't really fit currently in the incomplete block story. It's like a different approach. So the idea is, if you just look at the green plots, you have 3 treatments, and just ignore all the white plots, you basically have a randomized complete block design for the green plots. This is block 1, this is block 2, and this is block 3. So the idea of Federer's augmented design is that these 3 replicated checks would actually allow you to control for spatial variation within this block. So it says it augments the checks with other entries. You see, you have a large number of entries. You can see, I think it goes from 1 to 36, so you have quite a large number of entries that you are fitting in a limited number of plots. So the idea is here, again, sacrificing comparison between treatments, just to be able to put more accessions into your trial. Now, I thought this was pretty clever, but the idea, this is actually a real design used in UCLAC. Then the group of Brian Cullis came to the idea of what if we were to not use checks at all and actually use accessions and replicate those, because those checks, we're not interested in check performance. We're just going to use actual accessions and repeat those several times to get an idea of spatial heterogeneity over the trial, and then fill the remaining plots with other accessions. And then what your accession, the number of seed availability, would more be the determinant of what will become the replicated. So these are known as partially replicated designs. The colors just mean the level of replication. So every time it's like going one step further, sacrificing the comparison versus having more plots available for more accessions. It doesn't mean that you have to use p-rep designs, just to be clear. It means that if you have lots of accessions and you're willing to go to screen them at low precision, you go for this design. If you have a limited number of accessions and a huge amount of trial surface, you go for alpha designs, for example, or row-column designs, where you have full replicates, but still row-column effects. So there is no standard solution anymore. It very much depends on your needs, on what is it that you're willing to obtain. Now, how do you get to these more advanced designs like the P-Rep design? Well, in almost all cases, they're referred to as cyclic designs. You wouldn't actually be going through a book, well, that would be one option, and then try to find a design that has exactly the number of treatments and the number of rows and columns. The odds that you would find a book that has exactly that are getting very slim, especially with a large number of accessions. So these kind of designs are generated in an iterative fashion. This is from the manual of one of the R packages from Digger, it's called, the package. And this is a kind of the way that they start doing it. They make an initial design just by randomly, so this would be your trial in rows and columns, and just randomly distribute the accessions, and then start dividing these in blocks in either rows or columns, and each time trying to find a way where, so it swaps accessions iteratively, so it's an algorithm that just starts swapping accessions within one of these blocks and sees if the accuracy, the efficiency of the design increases. So what is efficiency in this case? There are various definitions, but usually it has to do with the comparison between treatments. If I have treatment A and treatment B, and I compare them, what would their standard error be? And I do this for all pairwise combination and calculate a kind of an average standard error on comparison. That is one form of efficiency. And you try to find a design just by iteratively improving the existing design. You can add assumed spatial effects, like the plots that are near to one another will have a similar, so you can make assumptions during the trial design that you expect to be present in the actual field trial. So this is completely virtual, but these kind of software packages are quite crucial when you want to use these more advanced designs. So they're generated using machine learning techniques like simulated annealing, and in my experience, this is not something that you want to automate completely. In almost all cases, you have to manually verify the resulting designs, often restart the iterations, try a few times more to get a design that perfectly fits your need with a nice separation of replicated plots versus unreplicated plots. And these kind of tools, by the way, they fit all sorts of incomplete block designs, from the initial alpha design where I started with, to row column designs, to P-Rep designs. Can all be fitted, but of course it takes some tinkering and getting used to working with those. Now, if we're going into direction of these incomplete block designs, it generally implies fitting a linear mix model. There is a plus to this. There is a greater flexibility in modeling what is called a variance, covariance structure and dependencies between observations. Sounds very complicated, but basically, if you fit, like if you have a standard randomized complete block design and you fit and you do a linear an analysis of variance, all the assumptions are already made for you, meaning, and your data just have to fit the assumptions. But in linear mixed models, you can make several assumptions and check them during the analysis. So it's actually you who decides, as the analyst, of what you assume to be important. Is there an effect of the rows? Is there an effect of columns? Is there a correlation between the observation that is non-genetic? These are all things that you can assume or check with linear mixed modeling. So similar to the first talk where I said there is no standard model for breeding value estimation, the same goes for trial analysis. There is no standard model. So this is different than using the classical analysis of variance where you just say, push the button, go, and you get the means of your accessions. Disadvantages, shrinkage. I'll talk about that later on. But that's quite important. In my point of view, the shrinkage of predictors is a quite important property for use in genetics or breeding, but I'll come back to that. Disadvantages, model fitting is more cumbersome. cumbersome is maybe the bad word, but it takes some getting used to. It's again, it's a kind of a profession. It's the work of a statistician. That doesn't mean everybody can learn it, but it takes some getting used to. And what is missing, there is no standard procedures for pairwise testing of treatments. Like Tukke's honest significant difference test. You can do pairwise testing, but you would have to do some corrections and things like that. There's no standard procedure. That's the point. Spatial modeling is one of the big advantages, I guess. Mixed model framework allows modeling of spatial effects. Most popular, but there are various different models. But this one is I guess, most easy to understand. So imagine that this is your trial, and let's just do the direction of the rows. So I'm just going to look at that. My matrix is a lot smaller than the number of rows here, so it just didn't have a picture that fitted nicely to this matrix. But imagine that you have a trial with 4 rows. The idea is that we're looking at the observations that were made, and the assumption is here that the non-genetic, so the residuals of the observations, so the part that is not related to genetics, that the correlation between the first plot and the second plot is this parameter rho. Now, the correlation between the first and the third plot is the square of this parameter. So in other words, it gets smaller because rho is a number between 0 and 1. So take the square of that and it becomes significantly smaller. And the correlation between the first and the fourth plot is even more. It's even smaller than that. So what you have is a correlation between plots that decays over distance. And that actually makes perfect sense if you consider this, for example, that this is a tree line where there's shade. So shade is a non-genetic effect. It's a residual effect. So plots that are here will generally have a lower performance than plots that are here that are not in the shady part of the trial. Then you can imagine that if we're looking at the row direction, that the autocorrelation that exists between the first and the second will be strong. Between the first and the third will already be much smaller, because the distance is bigger and the distance between the rows are different. So if you take this is a kind of what they call the autoregressive autocorrelation level 1. So in general, this is much used in spatial analysis of trials, but on the other hand, it's not something that you by definition would fit. So it's just one of the many possible models for modeling spatial variation in the trial. Now, one of the more difficult, and this is not, there could be a lot of discussion about this, but how are you going to compare one model over the other? For example, in one model, you will assume this autocorrelations, like I've shown in previous slide, and the other model you say there's no autocorrelation, because there is no tree line, for example, so why would you bother? So at that point, you need something to say that this model is better than the other. Because otherwise, it's just fitting 2 models and getting 2 different estimates, and then you'd never know which one is actually the more accurate estimate. Now, there are various criteria, so that's where the discussion might be. But if we're talking about parameters that are for residuals, like the autoregressive parameters, one of the more popular parameters is Akaiki's information criterion or the Bayesian information criterion. Sound very complicated, but formula is not so important, but maybe the idea. If you look at a trial and you do the analysis, basically in a linear mixed model analysis, you will calculate how likely, if you fit a model, you say there is an effect of my genotype, there is an effect of my block, of my row, of my column, whatever your model definition is. And then you can calculate inversely the likelihood that your data will have been generated by that model. So that is a likelihood-based approach. You will calculate a number, because everything is based on Gaussian distributions. You can calculate how likely it is that your data originated from such kind of model that you're doing. So if you compare these two likelihoods of two models, the one with the bigger likelihood is actually the better model, because it's more likely that your data arrived from model A than from model B. So in that case, you would only look at, this is the L, the log likelihood, but that's the same. You would actually be comparing likelihoods to compare models. But there is one problem, that likelihood generally does not decrease. If I add another parameter, if I add the effect of the block or the effect of the column, my likelihood will increase. So every parameter that I add will add a little bit more. Either the likelihood will stay the same, or it will become a little bit bigger. So what this formula actually does is give you -2 times the log likelihood plus a punishment for the number of parameters that your model is using. So, now it sounds complicated, but the idea is basically very simple. You have, on the one hand, the likelihood that your model is the correct one, and on the other hand, you have the number of parameters. You want to increase the likelihood as much as possible using the least possible number of parameters. What is a parameter? If I say there is an effect of the row, then that is costing you 1 parameter. Then t would be 1. If I say there is row and a column effect, that's 2 parameters. So that is every time you get punished for adding parameters to your model. And then the discussions start, what is a fair punishment? And that is where the differences start between statisticians and people. So there's a difference. Akaike's information criterion says that it's 2 times the number of parameters that you're using, 2 times t. That is a fair punishment. And the Bayesian information criterion says, no, it has to be t, the number of parameters, times log n, the number of residual degrees of freedom. A little bit more complicated, but actually, if you're using restricted maximum likelihood to get your estimates, that's just a way to find the most appropriate model factors. The two are completely equivalent because you would have to always keep the same number of fixed effects. So there's a lot of discussion, but basically both approaches will get you where you want. Choose one and try to find the one that gives you the lowest criteria, because you see it's -2 times the log likelihood, so the smaller, the better. That's the only thing you have to remember. And you can only compare models that have the same fixed effect structure if you're using REML, which most everybody does. Now, like I said, if you're using a randomized complete block design or a similar balanced replicated design, you wouldn't go through the effort to do linear mixed models. You'll use an analysis of variance to see if there's an importance of your blocks. You can do pairwise comparisons. You can verify your assumptions. And then comes the tricky part. Are we going to do a spatial analysis or not? Because spatial analysis, in most statisticians' minds, is something that is set in the linear mixed model framework. Now, there are approaches, and this is a very popular one, the nearest neighbor approach, which is also one of the implementations, is Schwarzbach's algorithm. So there is a bit of an impression going. This is what I like to refer to as the kind of old school approach. There's nothing wrong with it. It's just saying that we're going to use a randomized complete block design, and we're going to correct for spatial trends with nearest neighbors. So this nearest neighbor approach, Schwarzbach's algorithm, takes you back end of the 70s, early 80s. Is it wrong? No, it's not wrong. Just saying that there are currently, 2019, more elegant methods, more advanced methods to do these spatial analysis. Now, to give you an idea, this nearest neighbor analysis approach, so we're trying to do again the similar spatial correction. Remember the autoregressive type of thing, that we don't have this in a randomized complete block design, simple analysis. So we're trying to do a correction that is a bit of a manual process. And this is what is called second degree differencing. So imagine that this is one row of your trial, and the V's are your responses. And the index of the V is basically the position. So what you will calculate for each plot, you will calculate the actual value that you measured on that plot minus the average value of its two neighbors. So that would be what is called a second difference. Second because it uses two of the neighboring plots. You also have approaches that use one of the neighboring plots. So, and then afterwards it gets a little bit more complicated, so I wanted to save you all that, but basically it does a linear regressions on these second degree differences and calculates the effect of those. Now, this is popular for currently today, with a lot of breeding companies still use this, mainly because it's implemented in a number of software packages, which are either freely available or are very common in the breeding context, which makes them kind of the standard approach for spatial analysis. Now, there has been studies, or at least one study, where I took this image from, showing that this kind of approach, it's not wrong, but it really implies a strong detrending with a higher loss of degrees of freedom. So basically, you're assuming always that the spatial trend is there, and you're correcting your data for a spatial trend that might not even be there. So that is called detrending, and it's called a strong detrending. So you might be overcorrecting your data. That is the risk that you're doing here. It's also a bit inconvenient when your data is missing. You can imagine if one of these plots is missing, how do you calculate then for the other plots? How do you calculate the plots on the sides, which only have one neighbor? So there are all tricks and things you can do to fix those things, but it's not that clean of technique as it seems to be when implemented in these software packages. So basically what I'm trying to say is it's not wrong, but it has a tendency for strong detrending, and it's a bit superseded by models that model spatial effects directly on the observed data, so without calculating these differences. Like the one I showed you with the autoregressive part. It fits directly on the data. You don't calculate differences between plots. Plus, apparently one of them missing, this corrects in one dimension. The autoregressive trick you can do in dimension of rows and columns at the same time. This is always one-dimensional correction. So yes, it works, it's fine, but you might be interested in looking at more recent approaches, I'll call it. Coming back now, so I'm now coming back to the practical side of matters. So this was actually a very, very, very short introduction to why you would want to use linear mixed models. So key things are you can do incomplete block designs, you can analyze them, and two, you can do spatial analysis if required. So mixed model approaches, the focus now lies on getting a good model fit. There are various options for spatial trends. This one I already showed you in the previous presentation. And one of these things here, every mixed model has two components, a fixed effect structure and a random effect structure. And so if you use any linear mixed model software package, you'll have to define which of the factors you will put in the fixed part and which part you will put in the random part. Now, there's a nice theory on top of that, so it should be an easy decision to make. If you put things in the fixed effects part, if you're interested in specific treatments. For example, if you're doing a nitrogen level test, so you have different levels of nitrogen, let us say you have 4 levels of nitrogen, you do a test, then nitrogen level would be a fixed effect factor. You're really interested in what is the difference between the 4 levels. You could say the same for in a breeding context where you have accessions, and imagine that you have a low number of accessions, let's say again 4, then in that case you would generally also put them in the fixed effect structure, meaning that all the levels that you're interested in would be in the fixed effect part, and you're interested in testing their differences between the means. Now, it becomes a little bit more tricky in the random effects part. is where you say the inference is about the population of treatments. That's a very hard way of saying you're mostly interested in getting an idea of the variance of the parameter of the factor. So that is the population. So you're not really interested in 4 nitrogen levels. You're interested in the population, all the possible nitrogen levels. So that's a very theoretic assumption, but the key difference is you're going to assume that there is a certain distribution to these effects, while here you don't assume any distribution. That's the big difference. So if I have 4 treatment levels, I will assume I will have no assumption on where they're coming from. They're not necessarily coming from a normal distribution. They're just 4 different effects. Here you will assume that your effects are originating from a normal Gaussian distribution. Now, why does this matter? Well, first, remember, these are easy to fit. We only need 2 parameters, the mean and the variance. But 2, you also have already, this assumption will already skew your estimates that you obtain. Imagine that your factor levels are really not originating from a normal distribution. Analyzing them this way, putting them in the random effects, will give you predictions that are perfectly normally distributed. So you're actually pushing that assumption towards your estimation procedure. So that sounds bad. Still, in most cases, if we're talking in a breeding context, my advice is to put genetic effect, accession effect, in the random effects part. And therefore assuming that your accessions, their phenotype originates from a normal distribution. And this is where, again, a lot of arguing is going on, so you can discuss and debate about that. But first of all, it allows you to introduce a variance-covariance structure, which is a complicated word for saying you can assume that there are correlations between these effects, which you cannot do if you put it in here. What is a correlation between 2 genetic effects? Well, if one is the mother and the other is an offspring of that mother, their response will be highly correlated. Now, if you put them in the fixed effect, there is no way to introduce that additional information that you have, that those are in fact genetically correlated individuals. Putting those in the random effect structure allows you to introduce, remember, the numerator relationship matrix, that similarity matrix based on pedigree of marker information. Put it here, not possible. So if you put it here with some kind of similarity matrix, again, you're kind of also pushing an assumption. You're pushing the assumption that the effects are correlated that way and that they have a certain normal distribution pattern. But the most important reason, I find, is the shrinkage property of the blob. Now, the blob is just your estimate that you get for a random effect. It stands for best linear unbiased predictor. It's like if you calculate the mean of observations, that is an estimate. If you put something in a linear model, then you would get a least square estimate, which is very similar to a mean. If you put it in a random effects part, you get a best linear unbiased predictor. It's basically a different word for the estimate that you obtain for each level of your genetic effect. Now, these blobs, contrary to fixed effects, have an interesting property that is called shrinkage, and I try to explain it here. This is actually a random regression, so it has 2 parameters, but that's not important. Imagine that these are just 2 traits. So let's say yield and earliness. Doesn't matter. And these are you graph the blobs versus the least square estimates. So least square estimates basically say the mean value. You put them in the fixed effect. Now, for each individual, each arrow is an individual. The start of the arrow would be the least square estimate. So if you would have just put it in the fixed effect, the end of the arrow is the blob. Now, if you noticed, all arrows are pointing to the same position, namely the center of this, well, center here, I think, more or less. So the average of the population. So what is the idea of this shrinkage? It has to do with uncertainty of the estimate. For example, this is a very high estimate in least squares terms, but it also has a very high standard error. Probably means there was only one observation or just a few observations. It's a very unreliable estimate. So in least square estimates, it ends here. In BLUP, as you have assumed that it originates from a normal distribution, it is very unlikely that it would actually be here. So it shrunk down to the more likely value here. And the more it shrunk down, it depends on how reliable the estimate was. So these guys have hardly ever had no shrinkage whatsoever, meaning their estimates are very reliable. At that point, least square estimate and BLUP are the same. These guys, and here, mostly at the outside, are very unreliable and are shrunk down to the average of the population. Now, why is this good? Because most breeders at first glance think, wow, this is really, so I have a good guy now, and then I put it through the analysis routines, and it shrunk down, and it loses its distance versus the others. Well, the idea is that if it would be, it's a kind of a protection against outliers, because every breeder knows that if you test your individuals, each year you will have some guys that look really astonishing and will perform really well, as you equally have really bad guys each year. So that's the same. Now this shrinkage method protects you of choosing each time these outlying individuals, because there is no certainty that their actual position is there. It doesn't prevent you from keeping this individual on, and next year you will see, is it actually there or is the estimate really here? In most cases you will see that the shrinkage effect is correct. And animal breeders are so used to this system that in most cases, if they have the first results in from, I don't know, results on a dairy farm, they see the BLUP, and they know it has been shrunk down, and they just wait until new data arrives and make the exact same graph to see if the shrinkage is reduced or if it just shrinks further back to the mean. So they're really used to that mindset of it's unreliable, so the estimate is corrected, and the more reliable it gets, the more it will exceed out of the population. In reality, according to the normal distribution which you've put upon your analysis. Okay. So, there are, as you can imagine, hundreds of ways of fitting models, linear mixed models, to phenotypic trial data. Now, this is just one way, the way I will try to show you directly in Progeno, to give you an idea of how it works. You have what I call candidate model fitting. First of all, you'll have your response variable. You have to choose which trait you're going to analyze. Secondly, you fit an effect for the mean of the population. That's a bit of a standard thing. It's just to have an average trial effect. If you have a replication, you will fit that generally also as a fixed effect, because you have normally very few replications, just 1, 2, 3. That doesn't allow you to estimate a variance parameter. And then you will fit your genotype as a random effect, as shown here, and a residual. And then you can add candidate random effects, incomplete block designs, row columns, and one spatial model, either no or an autoregressive in column and in rows. Now, statisticians in the room know that there are many, many, many more spatial models that you could try, but just to give you an idea, that already so this is the base model, this will remain the same, and then you're going to try to add one of these. But I'm going to show you directly, this is a trial, an alfalfa trial from Bernadette. I've already shown it also some results during the actual UCLAG meeting. So instead of showing it on slide, just for those, this is actually how Progeno looks like. Gives you an idea. I was going to give a little bit of a demo also during the first part of the talk, but that didn't work out. Just giving you an idea of the trial. So Bernadette. So it has 400 accessions, which apparently isn't true. There are 387 actually in there. But that doesn't matter. So this is the design. It's a PREP design. You can't read the accessions, but they're not that very informative. But you do see the different colors. So the idea is the same. There are no replications. There are no incomplete blocks. It's just one big field, but certain accessions, so have been replicated. And then the intensity of the color determines the number of replications. So the really dark guys here, I believe there are 5 of them, they have been replicated 6 times. And the slightly lighter green, those have been replicated 2 times. I believe there are 28 of those. But do you see the ideas? This has been generated, I imagine. I don't know if I did this one or Miroslav, it was you? Okay. But the idea, I imagine that this has been done with some cyclic design generator. Thank you for confirming. And you see that the idea is to distribute these replicated parcels all over the trial to get an idea of the spatial heterogeneity that it might be present. So this trial has a lot of plant height observations. As you can see, I don't really know what the difference is. I think you explained, Bernadette, but it's a bit irrelevant. I found a kind of interesting spatial trend here in plant height 2 measured on the 26th of June. So what I'm going to do is take all of this data and send it to the analytics module. Remember the visual part of the analysis. So I have to give it a name. I'll just leave the default name. So I get the exact same data set. And let's just start with some visualization so you get an idea of what is actually in here. So I'll start with a flashy one. That's not very useful. So what I'm saying here is I want to have this as a column variable, that as a row variable, and then the z-axis would be the actual response variable. But it'll become more clear when you're actually looking at it. I should have done the other one. Okay, but this doesn't really say much. Sorry. Okay, so this plots my response, my plant height, as a kind of 3D surface over the trial surface. Now, you could hover over these and get an idea of who is where, but still I generally, while this looks quite flashy, it doesn't really give you a good impression of where there might be kind of spatial differences or if there are regions in the trial that are more or less prone to give bigger plant heights. So more interesting maybe is the heat map. So I guess this shouldn't be too hard to interpret. So you have your rows of your trial, your column of the trial, and the color just indicates the plant height that was observed. Now, again, open for discussion, but I feel like there's a bit of a trend here that this side of the trial has higher plant heights than the bottom right side, and especially the lower row, for example, as you see, smaller plant heights. Now, this could be related to the genetics, of course. It might be by accident that the low-performing height plants are actually here, but usually if you see these patterns, they're probably not genetic. So what we can do now is fit a mixed model to this data single trial analysis. The response variable is this measured 26th of June. UCLEC is my identifier of the genetics. I don't have a fixed replication. I don't have incomplete blocks. I do have a row, and I do have a column design. And I'll analyze this just as an example. So this is the output of the linear mixed model, and it doesn't give you really much. It gives you an idea of, first of all, the log likelihood. Remember, that's how likely is it, but this is a number that doesn't really mean much, right? It's a minus 912. It doesn't really give you much to go on. So these are all relative versus another model. So this, now I only have one. So same for the Akaikis information criteria. Remember that it was minus 2 times the log likelihood plus the number of parameters that I've used, the number of variance parameters, which is in this case 4, and they're mentioned here. So here I get an estimate. Look at the component part. The residual variation is 7.7. UCLEC ID, so that's my genetic part, is 14. So roughly speaking, this is contrary to your expectation, quite a good genetic response. It's the size of the genetic variation is twice the size of the residual variation. You can see this is what gamma expresses. So you get a good differentiation. You also get a variation of the row and a very small variance for the column. So is this a good model or not? Well, who can say? It doesn't look bad, definitely. But so one of the things that you can do now is add this, for example, the spatial part, the autoregressive part, remove the column, things like that. So that is basically what you could do, and you can have this done automatically for you. Because if you say that you agree that you will use AIC as a decision criterion, you could just ask a routine to just fit all possible candidate models and give you the one that has the highest, the lowest AIC value. So that is actually what this single trial model search does. You have plant height number 2, UCLEC ID, I don't have a rep, I don't have an incomplete block, I have a row and a column. I will extract the blobs because I'm so confident that this model will be the superior one, and I will extract the residuals. So this will take a little bit longer because now he's running over all different models. I can actually, this is running on my laptop, which is not so very beefy, but you can see it's using multiple processors at the same time. So it's trying to maximize the CPU that is available, and this is not by accident, arrives at the exact same model that I just fitted manually. So maybe not a very nice example, but showing you that now you know within those confines, of course, I just tested a limited set of possible candidate models. You could extend that set and put a lot of more spatial models in there. But with the game rules that I've fitted in here, this is the best fitting model. And interestingly, for example, I get now a new data set for each entry, an estimate, that is this blob value, and a standard error, giving you kind of a confidence level on that estimate. Why is that important? Well, we'll try to visualize. Remember that we're working with a P-rep design. So I'll just use a scatter plot, estimate, standard error. Go. You get a weird looking plot like this. What does this mean? Well, on the x-axis, the horizontal axis, I'm looking at my estimate, my blob. So the more to the, well, I don't know if high plants is something desirable, but imagine that it is, that you want the biggest blob, then you're looking for the guys that extend the most here to the right. However, you see that on the vertical axis you have the standard error, which you want as low as possible. Now, why are these guys here? Anybody? There are 5 of them. So these are the guys that have been replicated 6 times in the trial. These are the 28 guys that have been replicated 2 times. And these are the most bad estimates, those that are unreplicated. So you're not escaping from that by fitting linear mixed models. Estimates, no replication means, or limited replication means that you have high standard error on your estimates. Residuals, have I explained what a residual is? Residual is if you have your original phenotype, you take away the effect of the genotype, the effect of the row, the effect of the column. Whatever is left, means that it's not related to genotype, not row, not column. It's something that we cannot explain. You hope to see no spatial trend in your residuals because that should have been filtered out by my model. So I have this data set now called residuals. That's my input data set with one additional column called residual. And if I visualize that one, I will just use the heat map for comparison. Column, row, and color by residual. You get, I don't know if you remember the previous plot, but there you get a D-trend. Remember the bottom row, which was almost nearly completely blue, lower performing. Now these effects have been filtered out. You also see that it's not 100%. It depends a bit on your, so every spatial correction will put you to a certain amount. If you go further in it, like fitting those autoregressive, you would be able to filter that one out as well, but probably be overcorrecting. So that is the balance that you're trying to find. Comparison, remember what I said about this nearest neighbor kind of spatial detrending will always detrend very strongly. So here it's up to you to find the right balance. So that's what I said here. Yep, okay. Is it? Yes?
Do you use the replicates now, or do you use everything?
You use everything, but the replicates are of crucial importance to get an idea if they're row or column effects. Because imagine that if you have a row where there is not every plot in that row is unreplicated, then there is no way of knowing what is the effect of that row or what is the effect of the genotypes in that row. By having replicated plots within a row, of course they're replicated on other positions, you can distinguish between effects of rows and effects of genotypes. So you use everything, but without, if you wouldn't have any replicates, you wouldn't be able to do any analysis.
I have one question.
Yes?
How to prevent from overfitting in spatial modeling?
Overfitting and then?
Overfitting in spatial models.
In spatial models, okay. Well, to be honest, by using the AIC criterion, I don't often see overfitting. Because, for example, if I fit a model like I've tried here, you didn't see it, it happened in the background. It tried to fit autoregressive in both rows and columns. That costs you two parameters, two correlation parameters that had to be estimated. And in my experience, the punishment that you get for that in the AIC criterion is usually sufficient to rule out spatial models where there are none needed. Overfitting would only be a danger in my point of view if you really insist on fitting that without looking at the effects it has on the log likelihood. So for example, you see often in publications these really advanced spatial models where they fit autoregressive order 2 or 3 factors. So these take a lot of parameters, and I always wonder, has that been shown to be beneficial towards your analysis? So in my point of view, using one of these criterions with a correction, with a kind of punishment for the number of parameters, is a sort of protection against overfitting. Is it sufficient? That is hard to say, but
Yeah, Stephen, in this case, there was some block effects that were included in the design. So it's just your choice not to put them in the model.
The actual blocks, oh, I didn't even see them, probably.
Okay.
But there were no full replicates, right?
No.
No. There were smaller, incomplete blocks within the.
There are 4 blocks, in fact.
Within the rows and columns. Yeah, I'm sorry. I ignored them apparently. I'm guessing I'm again running out of time, so I'm just gonna. We have until when? 3:20? Okay. So you've seen this before, this hierarchical structure. So what I've shown you so far is an analysis on one trial and one possible approach to do it. Now, what I'm talking about here is what to do if you have multiple trials in multiple environments. So this brings you to the traditional discussion on G by E. Again, a topic where you could spend the whole afternoon alone talking about the effects, how to model G by E and so on, stability versus performance and so on. Now, I don't have the time to go over all these techniques, but just showing you, I'm guessing you're aware of what genotype by environment interaction is, right? So I can skip on that. One of these things I often encounter are ways of visualizing G by E or stability over, and one of these is Finlay-Wilkinson regression. Just to be clear, in my point of view, these are visualization, explorative methods. These are, in my point of view, not analysis methods. So don't confuse the two. This is just to get a feel of what is in this data. And the idea of this approach is to take the average of your environment, which is called here environmental quality. Imagine just take the average performance of each location. If each trial location is on the x-axis, and on the y-axis, you will put the average performance of each individual in each environment. Now, the idea is, imagine that how would the top performing, so each line, each color is an accession, is a genotype. How would the top performer look like? Well, a top performer would have a perfect horizontal line on 12. That would mean it performs in very bad conditions, and in very good conditions, it always performs maximum speed. It's the best thing. It doesn't matter if it doesn't have light, water, anything, nutrients, it will always perform. That would be your ideal variety. You can imagine that one has not been found yet. Now, how does a really bad one then look? Would be the other extreme, which would be something, I guess, just either stable bad, so a low line, always bad, doesn't matter what the environment is, or maybe a line that goes like this, meaning it's a bit good in bad conditions and it's really bad in bad conditions. So these are all weird situations. In most cases, you will find things like this. So the slope is an idea of stability. Now, like I said, if you have a flat line, but that is high up, that would be great. But normally you see something with a slope around 1, meaning that its performance will increase as the environment gets better. I guess that's a good thing. You have guys that perform bad, like the red one here. They perform bad under bad conditions, but they get a whole lot better in good conditions. And that then depends a bit on the goal that you're wanting to. So if you're looking at something that would work under marginal conditions, low fertilizer treatment, then that would not be your variety. So this is one of the explorative methods. Next one are, I guess, a little bit more complicated. I guess I don't have time to go into that, but these are ways of visualizing your G by E space as a principal component analysis. So very briefly, you fit a model that corrects for the genotype and the environment, and everything that is left, this part here, would be a kind of way to model genotype by environment. So this is a kind of a two-dimensional reduced space of your G by E space. G by E space would be a space where the environments are the dimension, and the performance in each of a variety would be the dots in that space. Now you reduce this to 2 dimensions, and you can look at each dot is an accession, and each line is an environment. Lines that cluster together, you know the drill. These are environments that are correlated. If you expand far away from the center, it means you have, for an accession, then you have a large G by E. If you project an accession on an environment, you get an idea of the G by E performance in that environment. Like in this case, this would be really high because the arrow is going that way. This one would be performing really bad in this environment. So that's a way to interpret that. I'll go very fast, and this is just a very similar approach, minor detail differences. But I would like to show you this before I end this presentation. So what I've shown before are visualizations of G by E. But it seems to throw overboard all the things that we did on the single trial analysis, meaning the spatial analysis, the row column blocks. We were just taking averages and putting them in regression plots or PCA plots. So the standard procedure, I guess, in most companies and institutes is to use a kind of a 2-stage approach to analyze multi-environment trials. So you would analyze per location, single trial, should present then the model specific for that location. So remember, like I did with the auto search trial, or with the auto model search, it will find the model that it fits best to this location, but for this location it could be a different model with different spatial structure or different row or column effect. So you do this for each location, so you get estimates for each location, corrected for spatial effect, trial effects, and so on. And this is what I call pseudo-observations. They're not actual, they're like averages, but I don't like the word. They're estimates by trial. And then you do a second linear mixed model analysis, if possible adding pedigree information to estimate the real breeding values. This is 2-stage approach. This works quite well, it's quite stable. Quite important, contrary to my statement that I like to put genotypes in as random effects, if you do this in a 2-stage analysis, it gets shrunk down twice. Remember the shrinkage? So if I put them in the first stage as random, and then again in the second stage, I'm shrinking them twice. The effect is called double shrinkage, obviously. So you should fit them here as a fixed effect in the first stage. Problems with this effect, with this way of working, is that there are various weighing mechanisms that say, imagine that you have a really bad trial, and the other one is really good. If I push them together like I've shown you in the graph, both trials would have an equal weight. So the bad trial would contribute just as much as the good trial. So that is not what you want. So normally you would cut down using some coefficient of variation or something. So there are various ways of using the standard error, the reliability of the estimates, and trying to add them into the second analysis round. Now that turns out to be quite difficult. If you use simple weighing models, you get an adverse effect on the estimates. If you use realistic weighing, so real standard errors, things become computational very prohibitive. So it's like you never win. Now, what I'm proposing here, I'll go directly to the graphical part and then end there, right, in case you're concerned. This is exactly the same. Brute force means I just go over all different candidate models, like I've just done during the demonstration, and I add all of these models into a single meta-mixed model definition. Basically saying you add all original data and you make a kind of model that encompasses all different trial models. So basically that means that you would have a separate residual variance in your model for this trial, a separate block effect for that trial, and so on. So you add all of these models together, and then, if needed, this is probably not needed, but adding G by E terms if needed, and do a single linear mixed model analysis on all your data in a single run. And this seems complicated, but again, remember this searching of single trial models is quite automatic. And then stacking them together in a single trial model is equally easy to do. So this kind of working actually allows you, and this is a kind of a study I made to compare those, allows you to be more efficient. Now, if what is shown here on this very unreadable graphic is that this is just a kind of simulation study, and each year you analyze the data either by just using this two-stage approach that I've shown you, or you use it with a single-stage approach that I've shown you in the slide before. And this is just the average Pearson correlation between the true breeding value and the estimated breeding value. And you see that using the single-stage analysis approach, so green and blue are just two different ways of doing that. The results are completely identical, so it doesn't matter. You see that they are, each time it is a higher correlation, meaning a higher accuracy using the single analysis approach versus two-stage analysis approach. Differences are slim, that is not so clear but on this axis, but the correlations are very close to one another. So it's not that it's highly superior, it's just easier to do. And secondly, it's always superior, although it's minor, the difference, but it will always be better than doing the two-stage analysis. Yeah, I'll end with that, I guess. Yes, I'll end with that.
Thank you, Steven. I assume there will be a lot of questions. Yeah.
Hi, nice talk. I just in the end, when you present the combined model, yeah, here. So what it looks like if you write the model? You actually write it. Do you just align the 5 other models, like you have XA, beta A plus XB, beta B, etc.?
Yeah, you're thinking about a formulation like in AS-REML or something that you would actually fit like the block effect. Well, then it would look like location A dot row effect, location B column effect. Mm-hmm. Sorry? It would be, in terms of variance, it would be a diagonal variance model indeed, yes. Your genotypes, of course, would introduce covariance between your observations of different trials, but your block effects and definitely your residual will be a complete diagonal block structure. But I'm not sure that is answering your...
I have no idea what it is, but...
Okay.
Okay, thank you.
In forage crop analysis and breeding, you often have many cuts per year, and so you get several data sets from the same plot design. How should we account for that in the models? Because they're not independent, but they are different.
Yeah, well, there are various approaches. And it very much depends on what you're trying to model. What is the trait? I know the trait is cut or dry matter yield, but what is it actually, the genetic effect you're trying to hunt for? Is it total performance? Because then you just sum and do a single analysis. Or is it the I don't know, the first cut? Or is it is it the growth curve over the year you try to fit? It very much depends on that, because what I've shown you so far were very basic models in terms of that indeed do not incorporate a kind of spatial trend with repeated observations over time. So you could add to this, specifically to the genetic effects, you could add to that, but generally, in my experience, these complicated models are not something you can automate, because you'll have to look if you can get variance parameter estimates, if your REML likelihood, sorry? Yeah, getting good starting values would be is of crucial importance, because REML is an iterative process, meaning you start with an initial value and try to improve it. But if you have many parameters, which is generally the case in these timelines, you often see that if your starting values are not in proximity of the actual value, that it doesn't converge at all. It goes all sorts of things. So these kind of models take manual curation. There is little to no automation in my experience.
More questions? Yeah.
What to do with non-normal traits? Because in our data there is many non-normal traits. If you try to fit the models, you will observe zero heritability for count traits and for ordinal traits.
Yes, that is.
With such traits, no bluffs, no prediction.
That is true. It's one of the reasons why I was enforcing on using objective scoring and objectively obtained phenotypes versus scales and binary traits like diseased, non-diseased, because it introduces all sorts of problems. And you are probably well aware that there are generalizations of the mixed models, but then you are equally aware that getting these parameters to converge and interpreting the results of these generalized linear mixed models is quite a challenge. So if it is me, but I know that is not your opinion, I generally try to transform the data to something that, exactly, I'm going to say it anyway, to transform the data to something that at least looks close to something that is normally distributed. However, if the information is just not there, and that is very often in binary traits, for example, then there is nothing you can do. Then it is, like you say, the heritability is zero, then you're done analyzing. So, but I try transformation. I'll say it again.
But in binomial distribution, no mean. In Poisson distribution, there is only mean. So we cannot translate mean and standard error to...
I know, like Poisson, yeah, but like these generalized linear mixed models, like a Poisson distribution. Yeah, if that is your opinion, that's...
Okay, another question? No? Okay, we're going to have a break.